Now let us study the structure and function of different classes of antibodies, which can be either IgG, IgM, IgA, IgD, or IgE. So here by classification we mean the different isotopes of the antibodies. IgG are antibodies which have gamma heavy chain, and when we look at their structure they are generally in the form of a monomer having one tetrameric structure, and this IgG can be further subclassified into four subclasses called IgG1, IgG2, IgG3, and IgG4. We will discuss them in our next slide. So IgG represents about 80% of the serum antibodies. So they are the maximum in number. So these IgG can be present in the blood, in the lymph, and in the intestine, with a half-life of about 3 days in serum. So IgG perform various different functions in an immune system. That is, they enhance phagocytosis, they help in neutralization of toxins and viruses. IgG are the only antibodies which can cross placenta and provide protection to the fetus, and it also helps in complement fixation. And IgG are antibodies which provide long-term protection because it can persist for months and years in the body. Here you can see the different subclasses of IgG like IgG1, IgG2, IgG3, and IgG4. So these are generally very highly conserved, and but they differ in their constant region, particularly in the hinge region here uh, and the upper CH2 domain. IgG1 is the most abundant type of immunoglobulin G, followed by IgG2, IgG3, and IgG4. So here you can see there is a difference in the number of disulfide bonds in the heavy chain among these different subclasses. So this is one of the differences which occurs in between these subclasses. IgG3 has the maximum number of amino acids in the hinge region, that is 62, followed by IgG1 which has 15 amino acids, and IgG2 and IgG4 have 12 amino acids in the hinge region. IgM are generally present in the form of a pentamer, that is, they are composed of 5 monomers of IgM which form an arrangement called pinwheel array, in which the Fc region is in the center and the Fab region is towards the periphery. The individual Fc region of each of the monomer is joined together by means of a chain called J chain. So this whole pentamer structure has 10 antigen binding sites or paratopes. That's why this IgM has a valency of 10. So IgM contributes towards 5 to 10% of the total serum antibodies, and these IgM can be present in the blood, the lymph, and on the surface of B cell as B cell receptors, and they are generally in the form of a monomer instead of a pentamer. So the half-life of IgM in the serum is 5 days. So remember, IgM are the first antibodies produced against any infection. So what they do, they result in elimination and agglutination of microbes. They are involved in complement fixation, that is via classical pathway. And the important thing is IgM do not cross the placenta. Here you can see a monomer of IgM, which is composed of 4 constant regions of heavy chain, as compared to IgG, which has only 3 constant regions of heavy chain. So each monomer is joined together by means of a disulfide bond apart from a J chain, which connects the whole structure to form a pinwheel array. So as you can also see, there are 10 antigen binding sites: 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 in a pentamer of IgM, giving it a valency of 10. IgA is composed of alpha heavy chain, and when we look at the structure, they are composed of a dimer, that is, it has two monomers of IgA joined together by a J chain. So this IgA can also be present in form of a secretory protein, so they are called SIgA or secretory IgA. So this IgA contributes towards 10 to 15% of the total serum antibodies, which may be present in secretions such as tears, saliva, intestine, and milk. Apart from that, IgA can also be present in the blood or the lymph. So in serum, IgA has a half-life of about 6 days, and it can perform various different functions. Like it can provide localized protection on the mucosal surfaces, and it can provide immunity to the infant digestive tract through the milk of mother, and it is also involved in alternate complement fixation pathway, but it cannot cross placenta. Here you can see a dimer of IgA. If you look at the monomeric form of IgA, it's composed of light chain and heavy chain. And heavy chain is composed of one variable region and 1, 2, and 3 constant regions. So these monomers are joined by a J chain as well as a secretory piece. The secretory component wraps around the two IgA monomers, giving it this form. And one function of the secretory components, it protects the IgA antibodies from degradation by the gastric acids and enzymes of the digestive system. So this property is especially important in the transfer of immune system components during breastfeeding.